Clinical side, yep. I guess we've had a good share of lots of molecules and pathways and signaling and all of that. And so the question is, how does it apply, and what does it look like? What does addiction look like in the clinical setting, and can we begin to look at neurobiology in the clinic? So it's really a pleasure to be here. It is my first time at CSL, so it's really nice. It's a lovely spot. We've talked quite a bit about the dark side, some, I guess, about the dark side with George Koob, and a little bit from Charlie as well. And I want to broaden, sort of take a step back with me and broaden the context of stress and the dark side. So we'll focus a little bit on that. The other piece that's really important, I think, it's nice to follow David Goldman, because he talked a little bit about the issues around phenotype, context, and disorders, and so we'll get into that a little bit as well. So what are the big questions that we've been sort of focusing on in my lab, and I think that are relevant overall? You've heard some of this. Of course, the $1 million question, I guess, is, why do people get addicted? And does stress have anything to do with it? So that's an important one, I think. Level of drug exposure. We've heard quite a bit about this in terms of neuroadaptations and development of compulsive motivation. So does that sort of suddenly happen when people become dependent, or is that a process of being exposed to drug? And if so, can we begin to characterize that and catch the early signs of compulsive motivation? So that would be one question. One big problem in the clinic, and I don't think we've talked much about reinstatement and relapse in the animal models here, and I don't know if anybody else will. I'll show some data from Yavin Shaham, who's a friend and colleague who's done a lot of work on the reinstatement models, and really how we are dealing with the problem of relapse and addiction. So why do we have high relapse rates? Why can't people just give up using drugs? Or how many people here have tried giving up a habit? Coffee. Everybody has tried giving it up. How many have been successful? Not coffee, or giving up coffee? Okay. So giving up habits are not easy. There are lots of issues related to that, and obviously several people have written about the fact that you can think about addictions as habits and bad habits. And so there are problems with that, and the question we got into quite deeply early on in my career was, can you really make any sense of the relapse rates and what leads to them? And if you can we begin to identify, is there a science behind it? Can we identify biomarkers and then bring that right into the clinic and see if you can see who are the people who are going to relapse early? So I'll talk about that. And then if stress in fact has effects both on the risk level and once you're addicted, and you again heard about that from George and Barbara Mason about treatment development, I would like to sort of, it's similar but also take a little bit of a step back on that and see, is there a different model or approach that we would take in terms of treatment development? So those are sort of the 4 questions, so there'll be 4 parts to the talk, and if we run out of time, we'll take off one of them or whatever. So lots of things that give us pleasure in the world, right? We've got our cigarettes and ice cream cones and Starbucks coffee and cocaine. I often get to tell people, often look at that and say, really, are those cocaine? Look like rocks, or, yeah. Anyway, chocolates, cheeseburgers, and so on, gambling, IV drug use, alcohol, all of that. So why aren't we all getting addicted? We all love, I mean, our reward centers are going to get activated. We all feel reinforcement and pleasure. And there's a lot of science of addiction that really, I mean, my humble perspective is we perseverate on the response of the accumbens to a rewarding substance, or the response of the reward pathway. I mean, it's called the reward pathway, and we'll talk a little bit about how the reward pathways are not really only the reward pathways. So we're sort of focused on that, and yet, of course, if you look at addiction risk of the commonly used substances, a substantial number of folks who use the substance will not get addicted. So what's in column 1 here, this is from Epidemiology, an old 20-year-old paper, but really Anthony has done a lot of work and lots of interesting data. The first column here is really just the prevalence of dependence for some of these most commonly used substances. But what's very interesting are the other 2 columns, which is this is not adjusted on users. So of course you have to start using something or liking something to start to even begin to lose control or get dependent. And so here's the proportion of users, and then what proportion get dependent. So this is one way of sort of thinking about what's the most addiction risk substance. And tobacco, it came up earlier today, is actually has the highest risk or addiction liability, so to speak. So if you're going to use tobacco among users, 31%, 32% will get dependent among users. But heroin is right up there, 23%. And relatively speaking, we've got a lot more people who have used alcohol at some point and users of alcohol, and 15.4% who end up meeting dependence criteria. And this is not current, this is lifetime, so at some point in time in their life. So this is sort of an interesting set of data and epidemiological information that's just good to put in context and in the back of your mind, because again, I think it highlights really clearly that there are significant numbers of people who do not get addicted. Other drugs overall, about 15% of the folks who are using, and here are the anxiolytics, which is everything together. And cocaine, the prevalence rates, of course, are small here, but the relative proportion is similar, and I just picked all the ones that go up close to the teens. So, of course, you heard quite a bit today already about genes and gene-environment interactions, and I'm not going to get into that, except that there are, of course, genetic and temperament factors. You've heard that, and what I want to focus on today are the bottom 2, which is stressful life events and adversity, impoverished environments, lack of support, maltreatment, cumulative stress, we'll talk about that, and level of drug exposure and changes in motivation and stress for drug, both of which we think may provide that context and phenotype, both regular clinical phenotypes as well as what we're very interested in, which is endophenotypes, particularly in terms of the stress response and stress neurobiology that may in fact then increase susceptibility and make up that potential difference of addiction risk. So we've had some references so far in the last few days about what stress is, and there may be stress of jumping off a cliff and how exciting that is, whereas perhaps being caught in a hurricane and really not knowing what to do. So different types of stressors. And so it's really important to just kind of, let's take a step back, define stress. And Bruce McEwen and several others have defined it. And from our perspective, in terms of studying stress, I'm less interested in the ones where you're jumping off the cliff because it's exciting and fun. And we'll talk about that. But if classic stress in terms of stress's effects on disease, why we care about stress is really, if you go look at that question, then the definition or the conceptualization that works for that are stimuli or events that are challenging, but the key elements, of course, are uncontrollability, unpredictability, and then being overwhelmed. And these will all, we'll refer to these in different ways. These can be mental, emotional, physical, or physiologic. And this is a little bit different because there's been reference again to stress as emotional things and psychological things. And from our perspective, we like to look at physiological stimuli as stressors too. Stimuli that are overwhelming the body, overwhelming the organism, pain, heat, Glucose, lots, very high dose glucose. You're going to see some, or lack of it. I'm going to show you some data on food as well, which we've become very interested in obesity and food. And I'll just show you one set of experiments. We're doing quite a bit with that. Now, of course, we really, really need our stress system, because increases in physiological and psychological stress is important in terms of adaptation. We'll touch on that. And in terms of survival as a species, we are the ones who are very good, in fact, at adapting to our environment among all other mammals. And I think the answer lies for a large part in the cortex and in how our brains have gotten wired over evolution. And then the key thing, two components. One is, of course, our response to the stress, and the second one is return to baseline, which we often do not pay as much attention to. And we will come back to this because it's relevant in addiction, at least in our hands, of what we find as changes in terms of neuroadaptations. So, what is stress? And this is what Charlie was talking about. Well, you know, you can jump off a cliff and you're well wired and it's really fun and exciting. And we just talked about stress being crucial for adaptation. And very quickly, I'm not going to review this. Everybody knows, I'm sure, that you get a activation of the stress pathways. There are two major pathways, the HPA, hypothalamic pituitary adrenal axis, and the autonomic nervous system and the adrenergic pathways, the sympathetic and the parasympathetic response. So you know that. We're not going to get into it, except that when we study stress in the brain, and people who are stress researchers in the brain often focus on one pathway and not the other, and not the interaction of the two. In the brain, there is intricate interaction of the two. If CRF is going up, you want to ask the question of what's happening to norepinephrine, or has norepinephrine contributed to that? So it's really important from our perspective to be thinking of both of those things. But in any event, so when it's mild to moderate levels, it's perfect. That's what we're wired for. So jumping off the cliff or writing a paper and it's coming together, you're working with a team, it's really challenging, but everybody's contributing, it's exciting, you have increased energy, you've got focused attention, you've got improved working memory and performance. My God, that's stressful, but my God, it's fun, it's good, it's exciting. So this is your first part of your U-shaped curve, and people talk about stress, arousal, and performance. But remember, in my definition of stress, would that meet stress? Probably not as much, okay? Because I'm not interested in, I'm sort of interested in that, but that's important for our learning and motivation pathways, absolutely. Learning and adaptation, but it's not interested in terms of health and disease. And if I'm interested in its impact on addiction and disease, you've done it successfully. You've adapted successfully. Great. The project works. The team high fives. Everybody's down at the bar drinking. That part I'm going to be interested in. All right. So there's enhanced performance, and so like I said, this is mild to moderate levels of stress or arousal, and you move on. You learn, and the next time around you have a project like that, you probably don't even get a cortisol response. You're just moving right along. And so what's our model in terms of what's going on in the brain? And how have we adapted to those mild to moderate levels of stress, or to even an uncontrollable level of stress? A lot of times we're focused on these two pathways, the HPA axis and the autonomic nervous system. We don't pay as much attention to what's happening in the brain in terms of stress and adaptation, unless, of course, you're a stress researcher. So this is a model that we've adapted from Trevor Robbins and Barry Everett in terms of what's going on in the brain. We've been working closely with Amy Arnsten, who's also a prefrontal neurobiologist. And together we built this model, and her focus has been very much looking at norepinephrine and dopamine pathways that are projecting into the prefrontal cortex and modulating the function of prefrontal neurons that are critical in exerting executive control, not just in terms of frontal functioning, which are important in helping us adapt and perhaps not get to that state of being overwhelmed and uncontrollable, but really in terms of also regulating each of these reward and limbic regions, like the amygdala, important in learning and emotional and motivation, hippocampus, the ventral and dorsal striatum, the thalamus in terms of the relay center and arousal, but also an area that we've gotten actually a pathway that we've gotten very interested in is our direct pathways from the PFC to the hypothalamus that are actually quite interesting and important, we think, in feeding and motivation for food, as well as perhaps for drugs. So we're sort of coming back to that. But what happens to the circuitry under uncontrollable stress? Oh, well, there's our little cartoon over there where when the prefrontal cortex is working and there's no stress, you've got this really good, all of these different, essentially, frontal functions are modulating the rest of the circuitry, and you've got good inhibition of inappropriate actions, regulating emotions, regulating the viscera, generally regulating the self and controlling impulses and all of that good stuff. But then if you have uncontrollable stress, we're just wired. We should just be totally, take into consideration the fact that as your level of stress goes up and becomes more and more uncontrollable, the PFC is going to start working less. Because why do you think that is? Why would you start to have a shutting down of the PFC with high levels of stress? Yeah. So what about an evolutionary explanation? If you're faced with a tiger, do you need to say, well, I don't know, it looks like my cat. I think it would be okay. Do you need your cognition at that point? No. You want to run. You want to get the heck out of there, right? And so just evolutionarily, there may be a reason why we're just wired to have this really important adaptive area of the brain that's key in control and regulation to be offline and start to not function as much in an acute situation. The problem arises that we don't normally get faced with a tiger in our regular day-to-day lives in the modern world. And in fact, our stressors are not acute and time-limited and short, even when they're uncontrollable. They tend to be longer-term solutions that don't go away. I'm thinking about child maltreatment. I'm thinking about divorce. I'm thinking about, you're going to hear about a lot of different types of stressors that impinge on us in terms of feeling overwhelmed, uncontrollable, unpredictable, okay? Remember that definition. We're going to keep coming back to that. And under those contexts, then, the question is not so much, well, the PFC went offline. The question is, how is the PFC struggling to sort of still continue to function and try to exert some sort of regulation and control when you're continuing to work and yet you've got these uncontrollable state that is overlaid over day-to-day functioning and performance. So the other key thing here to be focused on are that as a function of, and let me just go ahead and put this cartoon up, so if you have high levels of uncontrollable stress that are not in fact disappearing and you're not returning to baseline, over time then you might start to think about that prefrontal cortex being offline or beginning to show some level of atrophy and some disinhibition of these limbic and striatal circuits. Now, what's important here is the fact that there are adaptations going on in multiple systems that we were hearing about, especially the glucocorticoids, as well as norepinephrine, and GABA is highly sensitive to glucocorticoids. So a lot of the discussions in the past few days and in the talks were about GABA's influence on dopamine and its regulation of it in the ventral striatum. And I'm just going to ask you to take a step back and say, if you have an organism that has been under chronic stress or under uncontrollable stress with an adaptation where GABA may be down already, then what is the context of being exposed to drug? Okay? So you want to be thinking in terms of that, because this is where we have to get to if you want to think about your experiments and applying that in the human context. Your human context is not somebody who came in with the gene variation and now started using drug. Okay, that's not what's happening. I'm going to show you data in terms of how gets changed. Okay, and you might say, well, but stress effects are really more on the body and not on the brain. So let's just talk about very quickly. There are very clear data, and this is acute stress. This is not even chronic stress, but acute stress impairs attention, sustained attention, working memory, cognitive flexibility, goal-directed behavior, directly increases habit and habit formation, dorsal striatal effects, as well as medial PFC, and I'll show you some of that, increases impulsive choice and risk-taking. Just some of the critical executive functions that are important in, again, that regulation and control that I was showing you in the model. All right, so you want to broaden your perspective in thinking about bringing in this kind of context and phenotype in considering risk. Feel free to jump in, and if you have questions, you can absolutely have this be much more of a discussion. I don't know how that happened. Okay, so let's just take a step back and think about adverse childhood experiences, because of course you have stress prenatally, and we're talking about physiologic stress, and think about if you're using, as Marina was talking about this morning, tobacco exposure prenatally, alcohol exposure prenatally, is that a stressor for the fetus? If you're taking in large amounts of substance that clearly can be toxic to development, to neurons. And so in that context, I'm not going to be talking about prenatal exposure, but clearly that's important. Let's start postnatally and think about what we know of adverse childhood experiences. So what you're seeing up here, again, some epidemiologic data on ACEs, which is a very common acronym for adverse childhood experiences. You'll hear this. Filidi et al. Looked at one of the large hospital healthcare system databases and of close to 13,000 adults, and assessed rates of these adverse childhood experiences. 7 items only: witnessing domestic violence as a child, substance abuse in the household, family member in prison, mental illness in the household, sexual abuse, physical abuse, and verbal abuse. Unfortunately, as you can see, these are in blue are the ones from 1998, and you can look at the rates there. The prevalence of those are not low. Okay, so if you can see that number, the first one is 12.5, 23.6. I believe that's 3.4, 18.8, 22, 18.8, and 11.1. That's all in blue. The Centers for Disease Control now yearly is assessing adverse childhood experiences, because as you can see, these are unfortunately pretty common experiences. And in blue, in red here are the CDC's numbers from their morbidity and mortality report, again, assessed across five states, so large numbers of subjects. And why do we care? Because it looks like as the numbers of adverse childhood exposures go up, your risk can go up. And the way to break this down, and this is sort of interesting, these are not the same sample, so bear with me, I'm not asking you to do a direct comparison, but however, it's sort of interesting, to me it was, that over a course of 10 years, just randomly sampling the US population, the numbers of folks with no adverse childhood experiences was lower. Sort of interesting. Not sure if it's directly relevant, it's not the same population, but it was just interesting. And what was going up was 4 or more adverse childhood exposures. Okay, why does that matter? That matters because then when you look at the number of adverse childhood exposures and odds of risky health behaviors, and I took out the slide with number of ACEs and the odds of developing a chronic health problem. But the slopes are exactly the same. And the key thing here are ever used illicit drugs, right there, ever injected drugs, right there, and considering yourself an alcoholic, and so on. So number of adverse childhood exposures is associated with the odds of risky health behaviors going up, okay? What about cumulative adverse life events? And this is a sample between where the first cohort, it's about 2,400 in Florida, 2,400, maybe 2,200 folks being followed in Florida from the age of 15, 16, all the way on to 24, repeated over time. And the odds of developing, the probability of developing drug dependence goes up. Look at these curves. They're beautiful. In some ways, it's sort of, and it goes up based on the number of adverse life events they've been exposed to. It's by race, and a little bit, the slope was a little less steep for the non-Hispanic white Caucasian, but all the others are just sitting right on top of the others. So the magnitude changes, but really the direction is just beautifully linear in most ways. And then they've got nice data like this for other stress-related illnesses like other psychiatric disorders, as well as alcohol dependence and so on. So clearly, something is happening, and one is tempted to sort of say, well, maybe there are changes as a function of load, number of adverse events and load that increases risk in some way. This is a study that actually looked at the serotonin genotype, but what I'm showing you here is the main effect of stressful life events. Again, a longitudinal data set from Europe, I believe it was Europe, where children were followed ever since maybe age 9 or 10. And the main effect here in terms of low versus high is this, what you're seeing is binge drinking days. And yep, there's some effect of the serotonin genotype, but the more point here, the reason why I included it in here is that there's a main effect of stress, okay? So higher the stressful life events, exposure to current stressful life events, greater number of binge drinking days. So clearly something to be, certainly data that you cannot ignore, and perhaps we need to look a little bit more closely.
Can I make one comment?
Yeah.
That the relationship is the other way as well, that both maternal smoking and drug use and probably teenage binge drinking also increases the number of stressful life events. So the stressful life events definitely lead to the drug use, but the presence of substance abuse also increases the number and severity of stressful life events, which is a nasty side.
Yeah, and we're going to talk about that. And so that's part of what addiction will do to stress, and relapse is very much part of the plan. Yeah, thank you for that. Okay, so let's get into the brain then and see where is there scarring? Is there an impact? Is there a residue of these events? And if so, we're going to need to start worrying about it, right? So here is data from Richie Davidson's lab, where they looked at physically abused children and looked at, I think this may have been whole brain analysis, but I am not entirely sure. In any event, this is the orbitofrontal cortex, and what you're seeing is lower volume in the orbitofrontal cortex, which, if you know Nora Volkow's work, is an important area in the reward circuitry, and it is affected. It actually predicts their social functioning. Okay, so what you see here on the x-axis is social functioning, and volume is on the y-axis, and social functioning is worse with lower volume. Okay, so that's their data. These are work from Yale with Hilary Blumberg in collaboration with Linda Mays and our group. We looked at childhood trauma and its effects on whole brain volume. Now let me just clarify, because David, this issue came up in the last talk about masking and specific regions. Any imaging data that I'm going to be showing you today, there is no masking and there is no specific region. So I'm not asking the question, I'm only going to look at the amygdala and tell me what's happening in the amygdala. I'm going to ask the question, tell me in the whole brain. So you ask of the data, the whole brain, you do the corrections, and then you let that inform you. Okay? Just wanted to be clear. So if you have a question about that, it's not masked and it's not single region, it's whole brain data. So this is whole brain analysis, corrected, and then the regions that are coming up as related to childhood trauma are, of course, the amygdala hippocampus here, the OFC, but some clear sex differences. So some more of the striatal regions for the boys and more of the limbic and prefrontal regions for the girls. No effect of substance abuse here, okay, at this point. These are adolescents between the ages of 12 and 17. And their performance on prefrontal task, like the Wisconsin card sorting task, which is a executive function task of cognitive flexibility, requires some working memory, as well as some reasoning, and it's nicely correlated. The perseverative errors is correlated with the childhood trauma questionnaire scores, okay? So the children are rating their childhood trauma, reporting their childhood trauma, and this is an association with their performance on a cognitive task.
Is there some sort of critical period during development where like stress is most important? Because I know the SHRP is going on in humans like age 4 or 5.
Mm-hmm.
Is stress during that time?
Right.
Or like beyond that?
It's a really good question. There's mixed data on that. It's not really it's not that straightforward and consistent. So I think there probably are critical periods, but development is, you're sort of also going against just normal development as well in a different period. So it's not that clear cut. And I think there are some, like you're saying, there are some large scale studies ongoing, but it's not entirely clear. There was some evidence that may have been true with mood symptoms and anxiety symptoms and things like that. But particularly with cognitive function and prefrontal function, it's not clear.
Sorry, this was in teenagers?
Yes, the same.
So that's a time when that's changing
It's changing very
fast.
absolutely.
There must be a lot of variance there with age.
There is, but this is actually corrected for age. Age is a covariate in the model because it's 12 to 17-year-olds.
Okay. I think that people would develop very
Yeah.
differently in that time period.
Right.
You don't
So
know.
how much of the
Well,
variance
well,
does the
so-
covariate
I'm sorry?
explain? How much of the variance does the covariate explain?
So the way that you do this is the Wisconsin card sort is normed for the different age groups, so it's normed adjusted. Okay, so it's a very well-established cognitive test that's adjusted for age and gender for different groups. And so that's how you adjust for it.
But how do you covariate the amount of stress responsivity?
So it's not a biological stress response here. This is really their report of childhood trauma. Okay? And the childhood trauma questionnaire is a very well-established, reliable questionnaire on childhood trauma. You're going to see that maybe in another few slides. And there is the issue, of course, of it's still being retrospectively reported. Okay? And that is true of any kind of cumulative adversity and trauma. On the other hand, when you then look at that in terms of asking the same people those questions again, some corroboration with other sources, there's very high levels of reliability. And the alternative is that you go to the Department of Children and Family. There's a downside to that, because you only, unfortunately, the way our system is, only the extreme cases get reported, okay? So if you're really looking for concepts like stress load and variation and individual differences in response, you really need to find alternate ways, and that's why there has been quite a bit of work in terms of stress measurement and beginning to validate some of these other tools.
So where do you get your subjects from? How do you obtain the subjects for these studies?
From the community, different groups depending on who we are studying. These children were from the New Haven community, which is a mid-size, small to mid-sized city, urban environment. They came from the city as opposed to the suburbs, which is, so that's who they're. Yep.
How does, if you hear about childhood trauma, you have to report it. So how does that influence the study?
Right.
Unless you're doing it in adults.
Right. No, that's a very good question. We put in the requirements that need to be put in place for reporting. You can't get anything like that out of through your IRB. So you have to do that. But it's not, it depends. The one thing to and I did not show you the spread of the childhood trauma has 5 different subscales. It has physical abuse, emotional abuse, sexual abuse, physical neglect, and emotional neglect. Sexual abuse in this sample was very low. So physical abuse was reported, and then you make your investigations, you figure out, current, threat, all of that stuff, and we deal with what we have to do. Okay, This is a data set, again, adolescents, different group than the previous one. This group actually had children who were prenatally exposed to cocaine, but were not using themselves at the time of the scan. And this is a whole brain assessment of the association between stress and whole brain changes in terms of volume. And you've got superior frontal region and the precuneus region that are lower in volume related to stress. And that volume then predicts the odds of developing using substances. Early indication of beginning to use substances, okay? And those odds are pretty high. So for every milliliter change in volume, you get the odds of 0.7, essentially, a risk of going up.
So you mentioned that these had prenatal exposure to cocaine. Is there a control group without prenatal exposure
Yes.
to cocaine? Do you see that a similar?
There is an effect of the prenatal cocaine exposure per se, but not in predicting substance use. So the prenatal cocaine exposure has that group have a higher rate of sort of getting to using drugs. But overall, the volume predicting the drug use is not mediated by that. Now, there may be a sample issue there as well, but that's what we have right now with the sample. Okay?
What
Yeah.
kind of stressors are you using?
Again, this was really more stressful life events.
Oh,
Yeah, so I'll talk about experimental stressors as we get to that. Any other? Okay, so our working model about stress and addiction and self-control for a while has been, this was adapted, so ignore this part, which is sort of our link from stress, addiction, all the way to chronic disease, which is why we care about why people are getting addicted. But really, the key thing is that there are two pathways. One of the things we've proposed in the past is that there are two pathways with two separate risks going on. One is for stress, and the other one is for addiction, as you've been hearing. But in fact, or for drug use, in fact, they start to interact because stress has impacts on behavioral control, and that then starts to interact with escalation of drug use, and then begins to also affect the drug use itself will affect stress response, as Marina was talking about, and we'll talk about that in a little bit. And so then you get this sort of circle here where decreased behavioral control, stress-related behavioral control contributes to perhaps losing control more quickly, development of addiction, of course, adaptations, not just in the reward circuitry, but as Kub was describing in the stress circuitry, and you're stuck here. Meanwhile, of course, the less control you have, the less ability to regulate yourself, the more stress you're going to have, and the more quickly you're going to get to that overwhelmed state, which was our definition and conceptualization of stress. Okay, so this is really what these are sort of our working model, and neurobiologically I showed you what it was, which was all of those more complicated connections. So here's another interview that we use with our adults, which is called the Cumulative Stress Adversity Interview. The advantage to this is that it is a lifetime assessment of the bad things that have happened in your life. What I like about it, and what we're after in terms of the measurement side of it, is can we come up with sort of a loading? All of us have had stressors in our life, okay? And sometimes you sort of overcome some things, and other times you haven't. And so we all have some loading, which then may contribute to our risk for chronic diseases and to our context and phenotype interaction with the genes for chronic diseases. And so this is a 20-minute interview, a structured interview that's well validated. And we have now completed about 900 subjects have gone through this and all kinds of other phenotype data. And it has 4 subscales. And the first one is up there, major life events. So these are bad events: abandonment, divorce, separation, losing a child, parental substance abuse, relationship difficulties, things that are known, well known to have bad effects, okay, on health. The second one, life traumas, also bad effects: loss of home, witnessing being in an accident, in violent situations, all of the things that are listed there, being in combat, losing someone to violence, traumatic types of events, recent life events, past year, accidents, illnesses, all of those things, but focus more on the past year. And then the 4th subscale is this subscale that is much more your subjective distress, so your perceived stress. Now here is, we'll come back to that and to the question. Yep.
Do we know if there is a difference in people that have actually witnessed a violent episode like one of them that you described versus have lost someone hearing of it and not being physically there in terms of inputs and
Yep.
sensory set of stimulation?
It's a really good question. We don't know the answer to that, but people have done studies, for example, with 9/11. If you were right there versus you heard it, I mean, the whole country was traumatized at different levels from such a horrific experience. And it's, it's not really clear cut in terms of separating it out. My guess would be that there are clear differences. And clearly the level of being overwhelmed and feeling uncontrollable is going to be much higher, in the context of experiencing it. Yes.
I would extend that. What if someone close to you, like a child or a spouse or a relative, was in an accident or had a chronic illness? Does that also count as a stressful event?
Yeah, absolutely. I mean, I think it is a stressful event. It's listed somewhere up there. Yep. So anyway, these are bad events. What I like about this is you can separate out the impact of each of these. We are seeing actually differences in response, say, to longer-term abuse and trauma events versus recent life events on glucocorticoid response and morning cortisol levels. And this here is more your subjective state, so your perceived stress, which is an important element in stress, right? So if you're somebody that may feel overwhelmed more easily, or you have a negative emotionality in terms of trait, this is likely related to the serotonin gene and the depression and anxiety vulnerability idea, that you've got that negative emotionality, and that in addition contributes. Just an interesting measurement thing, these things, if I separate that out, or any perceived stress scale separate from events that have occurred, bad events that have occurred, they're not highly correlated. They're significantly correlated. It's about 0.5, anywhere between 0.4 and 0.6. So they are looking, or they can be separated, and therefore their impacts can be teased apart more interestingly. Yeah.
To follow up what Jessica was saying, there are many instances where the spouse of someone who's sick, and I see that in my office in Italy, with a long delay, they come back alone and say, you know what, I need help.
Yeah.
Because of stress, because of depression, because of anxiety, and things like that. So it's delayed. Sometimes it's not even a major disease, but it's a chronic disease.
Absolutely.
And that can really push them at some point over the edge. So it's very, very common.
It is. It's highly common. So,
Matt, you have a question?
I'm sorry.
This is really naive. Is there any correction that you make for positive life events? Is that even?
It's a really interesting point. People often will raise that. It's kind of like, well, how can you define stress? There's good stress, bad stress, all this stuff. Yeah, what we found, we measure it. What we find is that, of course, there's a lot of work now that's starting to happen on resilience. So one way of thinking about it is good stress or positive life events, sort of kind of like money in the bank, like you go through more positive events, somehow you're building up some resilience. And that's probably there. We're waiting on good data for that, especially in terms of impact on the brain. I will show some data on the fact that if you're doing, exercise, so if you're actually doing behavioral things, so not necessarily positive events like, oh, you had a child. Do people who have more children suddenly have some resilience built in? That's actually that's not true. But just to give an example, just wanted to get just wanted to get that out there. But certainly positive behavioral things that you can do, like exercise, obviously has positive effects, and there's several other things that we can talk about. Yeah.
So in figuring out the loading, I saw in there that you asked for age of first and last experience, and so like as time goes on, do you download stressors that happen a decade ago?
This is related to the point about developmental periods and things like that. We are not seeing that much of an impact of age in this cohort of 950 people. And in terms of the brain effect, which I'm going to show you right now. So just to take a step back, this should be not that novel for you guys. You've all seen the animal experiments. When I give human talks, people love this. So this is a neuron from medial prefrontal neuron. There's a control neuron, of course, a stress neuron. This is Radley et al.'s work showing that repeated stress changes synaptic density, dendritic branching, and all of the things that you can see here that show neuronal atrophy in one neuron. And so if you have, of course, the thousands of neurons that we have, you can start to think that overall then an impact of repeated stress on the brain would be what? If you had a number of neurons showing this. Bad, yes, bad. But what about quantifiably? You have a number of neurons that are shrinking, or what's that? Less gray matter. Okay, so you think we should be able to see that in humans now with neuroimaging? Okay, and here is data where this is, do not look for where there's shrinking or reduction. What the whole brain analysis is telling me here is these are the target regions after correction where there was lower volume associated with higher cumulative stress. So higher loading, higher the number of events, lower the volume. Where? Not everywhere. Prefrontal cortex, beautifully all over in the PFC, medial PFC, extending into some more rostrally as well, and ventrally, and then the striatum and the insula. Okay, direct association there between the cumulative adversity and brain volume. And then in the paper, we've also broken it down by the different subscales and looked at the impact of trauma, life trauma, major life trauma. Yeah.
How about studies where they've had twins and one went to combat and came back with PTSD? Another set of twins where one went to combat, didn't come back with PTSD, but they showed that the one that came back with PTSD, both of them had lower hippocampal volumes compared to the ones that didn't. So is there any other evidence also that starting hippocampal volume shows a greater resilience to stress?
Yes, so hippocampus, and I should have included that slide, I took it out, but the hippocampal volume particularly, now there's more evidence that it's not directly only related to PTSD, that early life stress, other kinds of trauma, you may not have the disorder of PTSD, but life trauma, which you can think of twins having, affects the hippocampus. And these are human studies. We, in fact, have a paper, which is what the data would be, which is whole brain analysis. It's really remarkable. It's whole brain analysis of a group of folks who don't have any psychiatric illness, community adults, but looking at trauma levels and childhood trauma levels. And you ask the whole brain, tell me where the impact is, and the impact is bilaterally in the hippocampus. Hippocampus, amygdala, I mean, you can now separate out the entire structure that is affected going into the parahippocampus. So it's it's a beautiful, you know.
So are London taxi cab drivers more resistant to stress?
We'll have to image
They have
them.
a totally different brain. They have the underground in maps.
Right, right.
Hey,
Okay,
I have
yeah.
a question about that. I mean, is chronic pain also gives you this
Yes.
loss? Is it the same pattern, or is it a different pattern you get with chronic pain?
In terms of brain volume loss,
Yes.
it's similar. I think
So
it
it's a
extends. It extends down, but
Yeah,
it goes down further into the limbic and midbrain regions. actually, but it's similar. I think the PFC is not as well. It's not that ventral. It's a little bit more in the dorsal lateral
Because
PFC.
that was pretty striking to me.
Yeah.
Chronic pain and you get this atrophy in the brain.
Yeah. But the prefrontal coming out is really amazing. I mean, it really, again, even in those studies, it goes all the way up too. Yeah.
So, and Rajita, the
Yes.
studies, for example, that they did with alcoholism, the shrinking, for example, in the amygdala was reversible. So, how much of this is reversible?
Right. Very good question. We're very interested in that. We're focused on that. There are a few studies, again, I pulled them out of here, but there are a couple of studies where you do start to see some coming back with, you have to do something, you have to do intervention. That's my talk about what do you do about the stress. We're not going to get into that, but there are ways. It's very early though, and we don't know how and where and how much. But clearly there are, and that's what's exciting too, because either medications or behaviorally, can you bring this back? It's not going to be instantaneous though. So we'll talk a little bit about that in the context of addiction. Okay, so can we induce stress in the laboratory? Of course we can. There are IRBs out there, so you are not going to be able to induce uncontrollable, really uncontrollable stress. So people have done things like standing up and giving a speech. You know, how bad is my cortisol looking right now? Is it really high as I'm talking to you? Well, you can, so those are called standard stressors. You can make stress people, but there's lots of individual differences. If I've done 100 of these, maybe I've habituated to it or whatever. So then there's, of course, cognitive challenge. Well, smarter people may be different in terms of how challenging a cognitive task is. So there are issues with standard tasks. There is a well-defined task called the Trier social stress test, which is about socially stressing somebody, having people not just do a speech, have them do a speech in front of people who are wearing lab coats and they're going to be assessing them, and have them do backwards counting while these people are judging and evaluating them, so you have three-person judge. And cortisol beautifully goes up. So if your end point is cortisol with the Trier social stress task, you're going to get a really nice effect using that, and that's a standard stressor. Our problem was that if you want to induce mood or drug craving, the social stress task doesn't work well. You suddenly don't get craving because it's a pretty contrived situation. My addicts, when I was testing that out and ruling that out as a tool to use, would say, "What are you doing? I don't really care about this." Yeah, whatever. Okay, I'll say something, but I don't care. I don't care who these people are either. You know, so they were way beyond all of that. They couldn't be bothered with this kind of stuff. You know, so it clearly was not working. On the other hand, what people who have been studying anxiety disorders for a while and wanted to do sort of in vivo exposure of anxiety and look at provocation of negative mood and anxiety have used this imagery method. It's a very structured imagery method. It's not like, you know, think about the last time you got stressed out, and I'll be sitting here measuring some heart rate and blood pressure. No, if I do that, you're most likely not going to be thinking about the last time you got stressed out. Who likes to think about aversive situations, right? You're going to think a little bit, and then you're going to think about other stuff. But this, in contrast, actually involves eliciting a specific situation, making up a script, and then playing that back, and relies on the fact that we are very we're imagery learners. We imagine things. We've got a powerful capacity for that. And then we have people imagine they're back in the context of those situations briefly. So I'm not going to say much more about the method because we could keep talking about it, except to say that it does work, and you'll see some data around it. Here I wanted to invoke it because that's how we expose people to stress, to a relaxed situation, which is an active control situation, as well as to a drug cue related situation. Again, you're going to hear about it. So I'm not going to say too much, except that I want to show you data on stress effects, functional effects on the brain with healthy folks. So this is community adults. So everybody got 2 trials of stress context, 2 trials of reward cue, and neutral imagery. I'm just going to show you stress because there was no differences, no effect of neutral on with cumulative adversity. So what you're seeing here is a spread of 75 people. and their stress response, but the association to cumulative adversity. Just like there was an association with volume, there is now an association with functional response to acute stress. So your history, remember the stress loading I was talking about? Stress loading is having an impact on your acute response to stressor, okay? And where? So clearly in the corticostriatal limbic regions here, your amygdala, hippocampus, insula, striatum. So as cumulative adversity goes up, as the number of events and your stressors have, stressful experiences have gone up, the activation in these key regions that are clearly involved in stress, this is your stress pathway and the reward pathway, are more susceptible to it. So it's cumulative adversity sort of sensitizing the acute stress response in the brain. Okay? Any questions about that? I just want to make sure we've got, okay. So key regions that you would expect are coming out. This is under revision. And then if you take your extreme of the distribution, just the top third and the bottom third, the top third clearly being the more vulnerable group, the PFC starts to come out. So the worst group in terms of cumulative adversity and exposure starts to show that even in that brief exposure of acute stress, the medial PFC going down into the OFC is going offline. Okay? Yeah.
So how old are those patients?
These people are adults, young adults, they're community adults recruited from the New Haven area coming in response to ads saying, yes, I want to participate in a study on health and emotions and whatever. So the medial OFC is lower in response to stress. Striatal and amygdala hippocampus is higher there. And so does that matter? In this case, we were looking at stress response, and so we said, well, does it predict the kinds of health symptoms? They were all healthy. Nobody had a disease. These are pretty clean subjects. But it turns out that the medial PFC and the hippocampus and the medial PFC were both correlated with their health symptoms. So aches and pains, the kinds of complaints, sleep problems, all of these different things that people have off and on were associated with the medial OFC and the right hippocampus, and the two regions were also connected. So thinking about it as a network, you've got a network here that's susceptible, that's clearly involved in stress, susceptible to cumulative adversity, so it's responding, it's getting sensitized to acute stress, and it's contributing to health symptoms. Okay, that's basically on the summary slide, so I'll move on. Any questions about that before we, okay. So I did say to you, I promised that we've got an interest in physiologic stress. What about the things that are going on in our body and the things we do to our bodies, like drug use and eating a high-fat meal or starving ourselves and things like that. So this is a paper where we've been collaborating with endocrinologists who use a hyperinsulinemic clamp to alter glucose levels. So this is an experiment we did where we had a euglycemic phase and a mild hypoglycemic phase. This is not clinically active hypoglycemia, okay? Glucose levels are really more on average about 68, and you can see that this is shaking. And our question was, what happens to motivation and preference for food as glucose levels are changing, okay? So while during these two periods of manipulation, people got to see pictures of pizza, ice cream cone, burger, cheeseburgers, cake, and broccoli and tofu and Brussels sprouts, and then some chairs and tables and lamps and things like that. Okay, so you had non-food pictures, and you had healthy food pictures, and you had high-calorie, high-fat pictures. And then they were rating their liking and their wanting, and they did this repeatedly, and these pictures were randomly presented. And here's your glucose levels. Like I said, it was really not in any danger zone. Nobody was having any clinical symptoms of hypoglycemia. It's just that your glucose levels are down, and if you don't eat and you're susceptible in some way, for say about 5 to 6 hours, you could go down to about 70. Okay, and this was about 68 to 70, which is where the mild hypoglycemia was. What was very interesting to us, that even with that mild hypoglycemia, plasma cortisol levels, which were measured right at the time, were increased. Okay, so there's your first sort of, here's a physiologic stressor. in some ways, and insulin, of course, was kept constant and was high to control glucose. So what happened in the brain? Overall, in response to the high-fat pictures, what you're seeing here in red and yellow is that euglycemia, the contrast, everything in the brain in the volt signal is always relative. Okay, you cannot get absolute measures, so it's relative to something. And here you're seeing euglycemic versus hypoglycemic in the red. So when it's red, U is higher. When it's blue, hypoglycemic condition, those regions and response is higher. Yeah.
So there is, as you know, Rajita, this new theory, this new movement about fasting for many hours. Mark Matson, the guy at the age
Right.
of 60, right? So basically the idea is that if you don't eat for several hours, you produce a certain level of stress in your body, which is releasing very positive factors, antioxidants, growth factors, and whatnot.
Yeah.
So he's claiming that
Yes.
it's a very positive, protective set of endogenous ligands that our body produces. What do you think?
Yeah.
So,
Well,
where's the Rubicon?
it's
Too
very
much
interesting. I don't think we know all of the effects, and we haven't imaged people, and we don't know. And I think there's probably going to be there are people who are resilient to effects, and then there are vulnerable people. So if you're somebody whose weight has changed back and forth, and you're doing that, and you're susceptible to food restriction effects in terms of the motivational pathway, which is what we know about that, that's not going to work for folks. But there is some we need to study it more. I won't go more into that, but it's very interesting. It's sort of it's curious. So anyway, what you see here is under euglycemia, when plasma cortisol was higher and glucose levels were mildly down, you have an increase in insula, hypothalamus, of course, putamen, increased reactivity, okay, and the caudate putamen, and reduction in the PFC, okay? So this means that prefrontal cortex was reduced under hypoglycemia, and it was higher under euglycemia. So that whole shutting down pattern I was telling you about with stress, with psychological stress, well, here's physiologic stress, and you see it beautifully. Lower PFC, higher limbic striatal response to these pictures, okay? And wanting levels went up. I think we have an image of that. Okay, no, next one. So this is just what you saw in the previous slide. We had mostly lean people in the sample, but there were 5 people who were obese, and that original sample, all that analysis was covariate with BMI. But if you break it down, this is very interesting to us that in fact the obese group was not showing increased PFC in the euglycemic condition. So they are seeing pictures of yummy foods. In addition, and this is I think the whole high-fat contrast. And the non-obese group are immediately bringing up and activating their PFC, but the obese group is not. In the meantime, of course, they are having much more bilateral activation of the limbic and striatal regions in response to those rewarding foods, high-fat, highly palatable foods. Wanting levels have gone up, liking levels have gone up.
So you didn't get any amygdala activation?
There is actually amygdala. You know, I looked for that, and there is.
Because I would have expected that.
Yeah.
based on the drugs.
Actually, not as much, but I thought we did have it. Not on these slices, but maybe it is. I'll have to check. And the paper has been published. It's in the Journal of Clinical Investigation in 2011. And so, of course, we're manipulating glucose. The question is glucose directly relevant here? And it is. Right here is the glucose response, and you can plot that with glucose levels, which we were getting every 5 minutes during that experiment. And right here in red is the euglycemic phase of glucose levels, and you see that nice association between the ventromedial PFC and glucose, which the prefrontal cortex utilizes really a lot of glucose. Yeah.
Have you looked at the opposite end of the spectrum, like people that are hyperglycemic, to see what effect that has on?
Yeah, we have not. We're starting a study. My colleague Bob Sherwin is doing a study actually with hypoglycemics and diabetics. But we've done some other work. We're really pursuing this very actively, and the PFC is going to be offline. I can just say that right from the start, which is really interesting because of other data, but I don't have it here to show it to you. Cortisol, on the other hand, was associated with insula and striatal response. So, of course, the Piazza model of glucocorticoids feeding right into the ventral striatum and perhaps having an impact on reward is something you can think of here. Okay, any other questions about that? So very quickly moving on to the next part, which is how does craving or compulsion build? So I showed you some part about compulsion with food. And the way I like to think about compulsion or our model really is one not that it sort of appears out of nowhere when you get addicted, but that desire and compulsion builds and that there's a development of it. And so we need to understand what are the factors that contribute to it. And if you just look up the Webster Dictionary, it's just important to sort of think about what are these constructs. Desire is to long for, hope for, express a wish, something that accompanies an unsatisfied state, okay, a feeling. Craving, then, you can move on and say it's much more of a longing, implying strong intent. With desire, you don't really have intent. With longing, you start to have intent. And longing and craving, you have intent. And you begin to facilitate an approach response, obviously related to that. And the compulsion, then, is irresistible impulse or urge, where you're going to do anything that might be better left undone or unsaid. Irrational urge to perform a repetitive action, that's much more of compulsive behavior. I think we're much more in addiction on the side of irresistible impulse and going ahead and acting. So if you do that, then you can think about, well, you know, one of you may say, well, you know, having a drink right now would be okay, and that would be a desire or wish, but you know, in the class here, you're not going to get up and go out there and go find a beer. Well, maybe you would, but maybe this would be a good time, right? But something is stopping you from doing that. And you could just do your own thought experiment and say, well, why am I not doing that? What's your assessment of your desire level? And if you start to move towards actually going out and getting it, well, something, you overrode something, right? You had to get out of the classroom to go do that. So we've proposed that really it's a dimensional idea, and in fact there's progression from no use all the way to binge-heavy independence. And so you might get on the post-addicted state over here with all of these changes that have been written about and you've been hearing about in terms of neuroadaptations. And one of the things that we're claiming, and I'll start to show you data on, is that when you're on this side of it, you're going to be somebody that responds to cues, but you have pretty good control, and you're not going to be stress sensitive to motivation. Whereas on this side, as you begin to progress, you're going to be stress responsive to motivation. And I'll talk a little bit more about that. Okay, here's an experiment that'll show you what I mean. Level of alcohol exposure and risk of alcohol abuse. Nobody in this sample is dependent, okay? We have non-smoking people, so it's not a nicotine effect that you're going to be seeing. We have light to moderate levels and binge to heavy drinkers. All functioning regular people. Two groups of folks, very well matched. Moderate drinkers, never binging. Okay, what's alcohol binge? Anybody know the numbers? What constitutes an alcohol binge? I love to do this in an audience because it's amazing how many people. How many drinks make up a binge?
5 or more.
All right, 5 or more for men, 4 or more for women. In any sitting episode, okay? 5 or more for men, 4 or more for women. Any one time. Yeah. That's usually the response I get. It's like, what? I could do 4. Anyway. So here, these are moderate drinkers who have never binged, all right? And these are binge and heavy drinkers who we can separate them out too. So binge drinkers, yes, I've gone above 5 or 4. How many times in a week or a month? Not weekly, perhaps once or twice a month. Okay, they would go into my binge category. If you're binging weekly, you're going to be moving into the heavy drinking because there's also heavy drinking criteria. What's that? 15 or more drinks a week in men. All right, heavy drinking. And 8, more than 8 in women in a week. Okay. So this is why level of exposure is important, right? If you don't drink, you're not going to have incentive salience. And if you drink it a lot, you're going to have more incentive salience. That's the idea. And of course, our groups beautifully break up on usual quantity per week. Here are the moderate drinkers, only 2 per 2. I'm sorry, this is drinks per occasion. It's 2 per occasion. closer to 3 to 4, and here they're in the 5 to 6 number of drinking days per week, and this is the one of the assessments for alcohol drinking and alcohol-related problems. So nicely divided up. We brought them into the laboratory, and we did an experiment, and I'm going to show you what basically they got exposed to stress, alcohol cues, and relaxing cues using our imagery method, 1 per day. So it's a 3-day experiment. This is in the afternoon. The IV goes in at around 2:30, and then there's actually goes in at 2, and then this is 2:40. So about 45-minute adaptation period. They have a 5-minute exposure to imagery, and these are the time points where we're making assessments. And then a tray comes out of beer, 2 cups of beer, and we do what is known as the Alan Marlatt alcohol taste task, and I'll show you that in a minute. But basically, this is context-based craving, so you change context briefly, and then you have context plus cues, and then, of course, you have measuring response, both subjective, physiologic, and biochemical. And here's the tray that comes out, 2 nice mugs of beer, 12-ounce beers, and a cup of water. This is the alcohol taste task, very well used in lots of different contexts. And what you're asking them is not something about stress or are you stressed. You're asking that separately. The tray comes out and you say, here's a task I want you to do. There are 2 beers, and the task is determine whether they're same or different, and drink as much as you want to determine whether you're same or different. Of course, we're looking at alcohol motivation and how much they're drinking to make that determination, right? But the task is go ahead and sip how much ever you want, and to make sure they're the same kind of beer or different. They love this task. People they're like, okay, how much can I drink? Can I have more? No, you can't. So it's really, in some ways, you can think about just your own response to how much 5 beers is one. People generally can hit ceiling in terms of consuming both. But the task is not to consume both. Task is really to taste it and say whether they're the same or different. Okay, was that? I went backwards. Okay, here's the data. This is imagery context, and this is when the beer tray comes out. And this is alcohol craving. Moderate drinkers, binge drinkers, and heavy drinkers, and you see clearly in the context of stress and drug cues, greater craving for the binge drinkers and the heavy drinkers. But what's going on here? Not so in the moderate drinkers. They don't have that much craving under stress and drug cues. There's some increase, but it's really not different. Here is where you start to see increase. So is there a sensitization of the stress pathways with alcohol? Alcohol actually, as does nicotine and cocaine, activates the HP axis very strongly, activates other extrahypothalamic stress pathways, as you've heard, and even at these levels, okay? So you don't have to be addicted to have activation. So you've got alcohol craving, but stress-induced craving starts to play more of a role in binge and heavy drinkers than in moderate drinkers. And of course, our manipulation is working, so you get really nice stress-related anxiety. What happens when the beer tray comes out? So now you're just adding to the context, of course, now even under the neutral condition, but much more so for the binge drinkers and heavy drinkers. So here's incentive salience for you right here. With the discrete cue of the beer, the binge drinkers and the heavy drinkers go up, even in the neutral context with increased craving. But what's happening here? Increases in anxiety. Nobody's drinking yet, right? So you've got increases in anxiety even in the drug cue context. All right, so this was stress. So under stress, you had anxiety. But here's drug cues. So just thinking about being at the bar with your friends, what's the alcohol cue context? I was celebrating my birthday and having several drinks. Positive situations. But now the tray has come out. If you're a binge and heavy drinker, you've got anxiety going up. Okay? And did you drink more? Yes, these people were drinking more. Very clear main effect of alcohol. A and B are just the 2 mugs. And how much alcohol did they consume? And clearly they were consuming more than moderate drinkers. So does craving matter? Did the craving response and the anxiety response predict how much they consumed under this taste task? And yes, it did. Increased alcohol craving among this group clearly predicted in the stress condition and in the neutral condition, but not in the cue condition, predicted how much alcohol they consumed. Okay, and of course, when the beer tray came out, the craving in response to both context and the discrete cue was much higher.
So how are you objectively measuring craving again? I'm sorry, I missed the point.
We have a visual analog scale that we use, as well as we have a 4-item, one of the alcohol urge questionnaires. So we use it we measured a couple of different ways. We've used this in multiple studies. So that craving is, what about cortisol? Cortisol among the binge drinkers and heavy drinkers was lower at baseline. Then here's when they started to drink. This is after drinking, meaning after the tray came out, and that's right here. And you start to see an increase in cortisol, more so in the heavy binge and heavy drinking. Of course, they drank more as well. And cortisol was negatively associated with amount consumed. Okay, so lower the cortisol, greater consumption, and that is really how the association has been reported in multiple studies. So it is a negative association, and that's because with high levels of drinking, you get desensitization of the HPA axis. So there's a downregulation, and you don't have, there's a flattening of the HPA axis. So generally, people have lower cortisol levels. Yeah.
Do you think would it work with other substances?
What will work? The
Yeah.
cortisol response?
Mm-hmm.
So with nicotine, that has been reported. With cocaine, people haven't looked at non-addicted folks, but with cocaine, it may well be that it's higher. I'll show you some data with our addicted individuals where actually what's predicting in the post-addicted state is the other way around. So it's very important where you're catching it. Okay? These people were then also imaged, a major subgroup of them, and here's the contrast among the social drinkers, everybody together, just looking at the stress condition versus the neutral condition. And in the stress condition, you get really nice midbrain activation, as well as PFC activation here, but not the medial, not the OFC and the medial PFC. There you've got a blunting, which is something I've shown you before. So under stress, we're seeing that. The cues, not so much, just really some dorsal medial activation there. But most of that is coming from the moderate drinkers. The binge and heavy drinkers start to show a blunting of response in the medial, in the PFC here, as well as in the ventral striatum. And what's associated with craving? Because they have craving even in the scanner in response to stress and alcohol cues like I'm showing you. And what's predicting the brain response that's predicting craving in the binge and heavy drinkers, again, non-dependent, is the ventromedial PFC and the ACC, the dorsolateral PFC. Blunted response, so lower response in the prefrontal regions, greater the craving. Okay? It's not the ventral striatum and other forebrain, midbrain regions. So stress and alcohol cue context and discrete cues each increase alcohol craving, significantly more so in the heavy drinkers than the moderate drinkers. It's also, that's the group that's consuming more alcohol, and stress-induced craving and discrete cue-induced craving is predicting that, and hypoactive PFC response is predicting the craving state. So we can move on. Let's see, how much more time do I have? We started.
So which comes first? That you drink a lot and you're more cue reactive, or you're more cue reactive than you're a drinker?
So these are data, the way we've shown it, it's a cross-sectional study, but the three groups are very carefully compared, carefully selected, without the effect of other drug use or family history and that kind of thing. And drinking, the level of alcohol exposure is clearly building incentive salience, is the way I'm interpreting this data. Meaning that...
But maybe the people who drink more, who are more vulnerable to drinking more...
Yes, of course. You could certainly have the fact, and that this experiment is not looking at that particular aspect. However, the data I'm not showing you and we are looking at now is what's the interaction between level of alcohol exposure and cumulative stress. Because the brain region that is showing the impact, which is related to your point here perhaps, are these regions that are affected directly by cumulative stress. I've shown you that. And yet here, in these folks, you're seeing lower activity under stress conditions is predicting craving response. So we're working on that now. Okay, So let's make a transition quickly over to, do we need a break? Are we good?
What's our 5-minute break?
5-minute break?
Just 5 minutes.
Yep.
It's a 5 minutes.
Sure.
How long have you got?
I can,
You've done 2, right?
I've done 2, but I can cut down. So I've got 4, but these 2 will go
Okay, well, that's
faster.
good. So maybe half an hour?
Half an hour.
Would that work?
Half an hour is fine. We'll stop wherever we can. Yep.
It's not